Ismail, and I am working as associate professor at the one of the largest public sector hospital in Pakistan. That is the leading hospital, and I am one of the TWAS affiliates and a member of the Asian Council for Science Editors and Global Health Security, and etc. etc. But today I'll be discussing on the mutational spectrum and in silico analysis of the mismatched repair genes in Pakistani oral squamous cell carcinoma patients. Please, next slide, please. So we all know that cancer is a disease that is characterized by uncontrolled cell division, and it is one of the major societal, public health concern, and it is now an economic problem because we know that a lot of those cancer and chemotherapy and treatments are very very difficult for now. We know that one out of six deaths are occurring because of the cancer amongst all communicable and non-communicable diseases. But because it is a non-communicable disease, so in that context, 1 out of 4 deaths are caused by the cancer, and it is one of the I'll just quickly go through. Can I have the next slide? These are some statistics only. So please go to the next. So previously, it was thought that cancer is a kind of a rich man disease. We know we were only thinking that it only affects the developed world, the European countries. But now, over the time, that narrative has changed. These are the statistics from the International Agency on Cancer Research and the GlobeCon, and you can see that Asia and Africa are the most prone continents to the cancer in the coming by 2025. You can see in Asia it will increase, the incidence may increase up to 64%, and in Africa you can see that it is around more than even 100%. Next, please. So, because my talk is on oral cancer, so just one slide about the introduction. That oral cancers, of course, are those cancers that originates in the oral cavity, and when that's related to the squamous cells, then that is termed as oral squamous cell carcinoma. The oral squamous cell carcinoma is ranked 16th in incidence and 15th in mortality. And like all cancers, it is characterized by the uncontrolled cellular growth, apoptotic suppression, and angiogenesis. And like every disease, early diagnosis would be significantly helpful. Next, please. And this is the estimated number of specific to the oral cancer. Previously, I shared the data of cancer overall of the all cancers. Now it's specifically related to the oral cancer, and you can again see Asia and Africa. So the point of putting these slides is that this idea of the cancer being only related to the European countries or to the developing countries because they are eating so many of junk food or something. So that is now challenged because we are more prone to the cancer than any other continent. Next, please. These are some Pakistani data, again from the GlobeCon. So you can see it will in future. It will increase to several folds. That is the projections. That is the data. So we, as a scientist and researcher, need to take care. It's alarming, and we should take care of these things. Next, please. Next slide. Same statistics. Just please go to the next slide. So let's talk about the molecular biology. Now we're coming to the scientific part of the this lecture. So we know that the cancer characterized the genetic hallmarks. So there are the proto-oncogenes, there are the tumor suppressor genes. These are there are the repair genes. When there are mutations in them, the divide uncontrollably. Let's say immortalization. These are all the cancer, and we our group is, dealing and identifying those mutations and hallmarks. Next, can I have the next slide? The which is by the way, the study is submitted to the scientific reports. What we do is to, that our objective is to, you are just moving too quickly now. We are. Can I have the previous slide, previous one? So our objectives is to perform the next generation whole exome sequencing of breast cancer as well as oral cancer patient from the formalin, fixed paraffin embedded blocks. Then we characterize those mutations. Then we study the impact of those mutations on the stability of the proteins. We also identify the interaction sites mutation. We want to know which mutation lies where in which important domains. We want to know the evolutionary significance of the mutations. We then perform the molecular dynamic simulations. We compare the wild and mutant type proteins. We then locate or review it for the biomarkers, potential genetic biomarkers, and then we associate those mutations, perform the association studies, and we in relation to the immunohistochemistry, clinical pathology, status of the cancer, and also there are other many social sort of factors, like for example hormonal status, whether some other risk factors like in oral cancer we are studying it in the context of using the tobacco, cigarette, etc. And then we further elevate it to identify potential targets for through molecular docking. Next, please. This is the bioinformatic pipeline that we are using. So when you do the sequence, you get the you get the raw reads. Then you have to generate the FASTQ files. Then there's the quality control. You need to be assured that your sequence is fine and it is an acceptable limit. The quality is sufficient. And then you have to use the BWA tool, the SAM tools. Then you generate use the NOR. Then you use the variant annotation tools, and then you eventually you have the CSV files that you use. So please go to the next slide. So this is the graphical abstract of the our study. It includes the right from the patient enrollment right to the analysis, from the simulations and all other. This is the graphical abstract. Please go ahead. I'll just now be presenting the results. Next. This is the demographic, the population characteristics. So in our study, male were comprised of around 29%, and the rest of them were the females. It's the age-wise distribution, the number of patients, and the patient history as well, whether they are taking how many of them are taking as the tobacco, how many they are smoking or something. So these are all the population characteristics, the cohort that we had. Next, please. This is we call it the lollipop plots. If you know that, so it shows that which domain has the mutations, and to what extent and how many mutations are reported per domain. So in this study, we have included 5 genes, all related to the mismatch repair pathways. So these are all this distribution. Next, please. This shows the exonic distributions of all those mutations. So this graph that tells us which exon is most prone to some specific mutations. Next, please. This is the predictions about the pathogenicity. So you have the all the different mutations that we got. Then we predicted their pathogenicity. There are some mutations, for example, which were highly pathogenic. Like for example, you can see the polyphen 2 predictions, you can see the SIFT predictions, you can see the mutation tester predictions, ClinVar, and all the other prediction databases that we are having and we were having access to. So all of these basically telling you which mutations were or how many mutations were highly pathogenic or kind of have the potential to cause the cancer. Next, please. This is the evolutionary the this is donut plot is referring to the conserved results. So it's another tool used to identify the evolutionary conservation. So here we can see that you have around all those mutations. If that is this is a dark purple kind of, so this was a highly conserved. And if on the other hand, if it was on the light green side, so this was the relatively non-conserved mutation. So these have their own interpretations. Either they are functional, they are buried residues, or they are structural residues. So we identified that. And furthermore, so you have these stability predictions as well. So you can see that on the table that I'm showing you, all of those mutations eventually has the destabilizing effect on the structure of the protein. Next, please. Next, please. This is the then what we do is to model those proteins, the mutant as well as the sorry previous one, previous one, please. The previous slide, please. So here what we do is to do all the molecular tools. We model the wild and the mutant proteins. We then superimpose them. You can see all those structures. The next one, the structural, the which is showing the structure. The next one. This one, yes. Please stick to this slide. And then when we compare the wild and mutant proteins, superimpose them, then we do the molecular dynamic simulations, and we want to study how much they are affecting the protein and in which context, in which parameters they are affecting the protein. So all these graphs are coming from the molecular dynamic simulations results. All these results are of the MDS simulation. Next, please. So one of the important results from the MDS simulations through GROMEX that we have done is to study the radius of gyration. Now, those who know a bit about the protein chemistry, so if the radius of gyration becomes enlarged or become increased, so the protein becomes destabilized. And you can see that we have compared the normal and the mutant proteins. You can see this the gyration radius has slightly increased to about few nanometers. So which means that all of the predictions were that the mutations have actually in real destabilized the protein structure. It has created more unfolded proteins as compared to the wild type. Next, please. This is the association results. So all of these mutations we study in the context of the clinical pathological parameters. We studied in comparison to different risk factors. So I'll just jump directly go to the next slide. These are the target results. It tells you about the when a mutation is there. And we compare it to the wild type. So how much is it affecting the genes in the surrounding? So you can see that the mutant graph is way higher as compared to the wild type of the same protein. So it again says that there is some greater effects of those mutations. Next, please. This is the pathway analysis that we have done. It's from the string databases and the gene mania databases. So we want to study which proteins or which pathways are kind of disturbed because of those mutations. Next, please. So this is the stabilizing effect, stability, or all those single nucleotide variants. So the frame shift mutation has a destabilizing effect, and we calculated through the differences between the energies, that delta G energies. Next, please. So jumping into the conclusion, so what we to cut everything short that we have done, our main finding was the one of the findings was the on the check one mutations that were present almost across all the patients that we were having. So, and then there was the TP53 mutations, P33R, in which protein changes into arginine, and the ATR mutations were reported in the 77 and 74% of the patients. So we believe that these mutations are recurrent in our population, and they can be used for an external. They can be used for the biomarker applications. And then one important finding was that TP53R43H, in which arginine converts to histidine, mutates into histidine, is potentially very pathogenic because all of the SAM databases predicted this mutation as highly pathogenic. Across the 42 mutations, all were predicted to have a destabilizing effect on the protein, and which was complemented by the molecular dynamic simulation results run through GROMEX. The associated results, one notable finding was that the TP53 P33R mutation, in which proline converts to the arginine, it was very common in NSWAR users. The NSWAR users were basically the tobacco, the people who use the tobacco. So these mutations were, present in all the tobacco users. And the patient with both germline and somatic TP53 mutation has significantly lower survival rate as compared to. So those patients which have germline mutations on the TP53 has a less survival rate as compared to the non-germline mutations. So moving ahead.
Yes,
Next
thank you,
slide.
Dr. Khalil.
These
Yeah,
are some of the
this is the
publications
last.
from
Okay.
yeah. This is the second last. So these are some of the publications from our work. Next, please. Next slide, please. Any questions? And then thank you very much.